This conference will now be recorded.
Hello students, good evening. I'm Dr. Divya. So this is the last class of our second chapter, okay? So we have already learned about pollination, and after that how fertilization is taking place, right? So all those things we have learned. Now we are moving to learn some important points. So you can expect more neat questions from this area. So the first topic is double fertilization. We are going to learn about double fertilization now. So fertilization, you know what is fertilization? It is the fusion of these gametes. Male and female gametes are getting fused, right? So that procedure or process is called as fertilization, right? And the process of double fertilization. What is double fertilization? From the name itself or the title itself, it's clear. Double fertilization. Fertilization is taking place 2 times. So that is called as double fertilization. And we'll see how it is taking place. And the process of double fertilization, it was first discovered by S.G. Navashin. And he did his experiments in Lilium species. He discovered the double fertilization process in Lilium species. No doubt. And you know that after pollination process, the pollen grains, that pollen grains will form a pollen tube. You can see here in this picture, the pollen grain, it will reach on the stigmatic surface, and from there it will form a pollen tube. And I have already explained the rejection and acceptance of this pollen grains and all. The compatible pollen which is reaching on stigmatic surface, that will germinate, and remaining pollens will not germinate. The compatible, it should be matching to that stigmatic surface. There is a chemistry between the pollen grain and the stigmatic surface. So there, so many chemicals will get produced, and there is an interaction between the pollen grains and the chemicals of the stigmatic surface. So as a result of all these chemical interaction, the pollen grains may get accepted sometimes, or it may get rejected also. It depends upon the chemistry between the pollen grain and the stigmatic surface, right? So after accepting, a pollen grain will germinate on that stigmatic surface, and it will form a pollen tube. You can see here in the first picture, you can see a yellow color tube which is forming. So that pollen grain will germinate and it will form a pollen tube. And this pollen tube will come out through the germ pore. This and all I have already explained in the last class. Through germ pore, that pollen grain will form a pollen tube. And that pollen tube contains 2 male gametes. We have already explained the male gametes. There is a 2-cell stage and 3-cell stage, right? So the pollen tube contains 2 male gametes. So all those procedures we have already discussed. So the pollen tube with 2 male gametes, each male gamete is, it's haploid or diploid, that male gamete, it is haploid. That male gamete is haploid, and the haploid 2 male gametes, they have 1 set of chromosomes. That's why they are called as haploid. They have n number of chromosomes, right? And this pollen tube with 2 male gametes enters to the ovary. Where it will enter? Here you can see it is entering to the ovary. Here you can see the pollen tube which is coming from the stigmatic surface. It is entering to the ovary. Through which region it is entering? Through the microphylla region it is entering. Through microphylla region, that pollen tube is entering to the ovary, and that is a common mode of entry. Okay, there are other modes also. Through other regions also the pollen tube will enter, that I'll explain. But in normal case, it will enter through the micropylar region. That's why the integuments are leaving a gap. That region, that entering region is called as micropylar region. So there is a gap. The integument is not covering completely. It is leaving a gap for the pollen tube entry. So that is a micropylar region, and through that region, the germinated pollen grain already that is releasing that contents to the pollen tube, and that pollen tube, it is entering through the micropylar region to the ovary. And this is the common method of entry. And in most of the plants, we can see this common mode of entry through micropylar region. Note down, through micropylar region, that is the normal. In normally almost all the plants, we are seeing this micropylar entry. And the pollen tube, first it will pierce through the nucellus. You know that here nucellus is there around this. So through that nucellus, first it will pierce. We have already explained. Here you can see it is entering through the nucellus. First it is piercing through the nucellus, then it enters to embryo sac. Here you can see through nucellus it is entering to the embryo sac. Right? Clear? So after that is entering to the embryo sac, and the tip of the pollen tube. You know, only that tip, whole pollen tube is not entering inside. Only that tip of the pollen tube, it is entering, or it is penetrating to the embryo sac, and it is reaching near the egg apparatus. You can see here the egg cell. So near to that it is reaching, right? The pollen tube, first it is coming through nucellus, then it is entering to the embryo sac, and only the tip of the pollen tube is entering to the embryo sac, and that reaches near the egg cell apparatus. You can see near to the egg cell that pollen tube is reaching. And after reaching, the pollen tube enters to any one of the Steiner gates. While talking about the Steiner gates itself, I explained pollen tube entry. That is, what is the function of the Steiner gates? They are guiding the pollen tube for the entry, right? It is helping the pollen tube for entering inside. It is guiding. There is some kind of chemical release from this, so that is guiding this pollen tube towards the egg cell. Then after that entry, that pollen tube, it releases the two male gametes. You can see here the releasing of male gametes. Here you can see at the tip of this pollen tube, you can see two male gametes. So the pollen tube, after reaching inside, it is releasing the male gametes. And there are two male gametes, right? Among the two male gametes, both are haploid. Both male gametes are in haploid stage. So the male gamete, among these two male gametes, one male gamete fuses with haploid egg cell. Among the two male gametes, one male gamete is getting fused with the haploid egg. Egg is also haploid. This coming male gamete is also haploid, right? So the egg and one of the male gamete, it will fuse together and it forms a, there is a haploid set of chromosome in the male gamete and another set of haploid set of chromosome in the female or the egg cell. So both will join and it will form a diploid zygote. So in the zygote will be in diploid condition. There will be two sets of chromosome, right? There is not a single set of chromosome. From the male gamete, one set will come, and female gamete, another set. So zygote is diploid, 2n number of chromosomes in zygote. So this is called as syngamy or generative fertilization. Note down syngamy, otherwise it is called as generative fertilization. It is the fusion of male gamete, one of the haploid male gamete and haploid egg cell. Those cells are getting fused and it is forming a diploid zygote. This is called as syngamy or generative fertilization. Here you can see all these things. And the diploid zygote is getting formed. The zygote is diploid, but both the gametes are haploid. Syngamy is otherwise called as generative fertilization. And there is one more male gamete, right? There is another male gamete, and these remaining male gamete that fuses with the diploid secondary nucleus. Secondary nucleus, you know, at the center, the secondary nucleus is present, right? So this second male gamete or the remaining male gamete that fuses with the secondary nucleus. Secondary nucleus means already it is in diploid condition because two cells are getting polar nuclei. Those two cells are getting fused and it is forming a secondary nucleus, right? So the secondary nucleus is already in a diploid condition. Then to that, one more set of chromosome from the remaining male gamete, one more set of chromosome it is adding, right? So that results in a triple fusion, correct? So to secondary nucleus, which is already in a diploid condition, another set of chromosome is adding. So that is a triple fusion or vegetative fertilization. That is called as vegetative fertilization. Syngamy is called as generative fertilization, and here with the secondary nucleus, another remaining male gamete is getting fused. That is called as vegetative fertilization. So this forms the vegetative fertilization or the triple fusion that forms a triploid primary endosperm nucleus. What is that? This forms a primary endosperm nucleus, that is triploid, that is in triploid condition because already the secondary nucleus is present at the center. To that secondary nucleus, the remaining male gamete is getting fused. So already the secondary nucleus is in diploid condition. To that, another set of chromosome is getting added. So it is in triploid condition. There is 3 sets of chromosomes. 3 chromosome sets are present in that. So that fusion is taking place third time. Polar nuclei, both polar nuclei got fused. First fusion, then this is the triple fusion, third time fusion, or the 3 sets of chromosomes are getting fused in that. So that is a triploid condition and 3n number of chromosomes. There is 3 sets of chromosomes in that. Okay. It is also called as primary endosperm nucleus. Don't forget primary endosperm nucleus. That primary endosperm nucleus is the starting stage of endosperm, which will give rise to endosperm in later events that will give rise to endosperm. So that is the primary stage. This P in short form we will call as PEN or primary endosperm nucleus. This primary endosperm nucleus later it will give rise to endosperm. So this is the primary stage. Got it? So the pollen tube, it is guided by the chemicals secreted by cyanogates present in the embryo sac, and it enters to the ovule sac through micropyle. Okay. So after entering to the cyanogates, 2 male gametes are released into the cytoplasm of cyanogates. One of the male gamete, the first male gamete, it will move towards egg cell and nuclear fusion is taking place. That is called as syngamy. And syngamy results in the formation of diploid zygote. After syngamy, the zygote formed will be in diploid condition. There will be 2 sets of chromosomes. 2n number of chromosomes will be present in zygote. Then the remaining male gamete, other male gamete fuses with the polar nuclei and produces a primary endosperm nucleus. This polar nuclei will there is already one fusion over that is forming the secondary nucleus, right? secondary nucleus, and to this secondary nucleus one more male gamete is getting fused and it is forming primary endosperm nucleus. And this primary endosperm nucleus is triploid. 3 sets of chromosomes are there, and this is also called as triple fusion. Here 3 fusions are taking place, that is called as triple fusion. Okay. Next is and here the process of fertilization. Why we are calling it as double fertilization? The process of fertilization occurs twice. 2 times the process of fertilization is taking place in the same embryo sac. See, I am talking about this embryo sac here. Fertilization, the process of fertilization is taking place 2 times at the same time by 2 male gametes, right? That's why it is called as double fertilization. So 2 types of fusion. First one is singamy and triple fusion. These both are taking place in the same embryo. So 2 times the process of fertilization is taking place. 2 male gametes. One male gamete will move towards egg cell and another one towards the secondary nucleus. So both sides fusion, 2 fusions are taking place. So 2 fertilization processes are taking place. So it is called as double fertilization. It is happening 2 times. And after triple fusion, the central cell forms primary endosperm cell. Very important. After triple fusion, only after triple fusion, that central cell, here the central cell will form primary endosperm cell or primary endosperm nucleus, that is in triploid condition, and this will give rise to endosperm. Later it will give rise to endosperm, and the zygote, egg and first male gamete that fuse to form the zygote, which is in diploid condition. That diploid zygote, it will divide and redivide and it is forming an embryo that will give rise to embryo. Zygote will give rise to embryo and primary endosperm nucleus will give rise to endosperm. Don't get confused. Here the center one, the primary endosperm nucleus will give rise to endosperm and zygote will give rise to embryo. Okay. Next. Here I said normally for fertilization the pollen tube containing 2 male gametes that enters to the ovule. Normally it is entering through micropylar region, right? After germination, when the pollen grain is reaching on stigmatic surface, it is germinating and that pollen tube is entering to the ovule. That pollen tube is entering to the ovule through the micropylar region. That is the normal case. Okay, with 2 male gametes. So that with the 2 male gametes, that pollen tube is entering to the ovule. And if the pollen tube is entering through micropylar region, it is called as sporogamy. What is sporogamy? The entry of the pollen tube through micropylar region. That is called as sporogamy. Here you can see in this picture, it is entering through the micropylar. This is the pollen tube, that red colored marked one. So that is the pollen tube. Here I have marked the pollen tube. So that is entering through the micropylar region. See, this is the micropylar region. The pollen tube is entering through the micropylar region. So that type of entry is called as sporogamy. So sporogamy is the common mode of entry of pollen tube into the ovule. Into the ovule, the pollen tube is entering through the micropylar region. That is called as sporogamy. Then pollen tube enters through the chalasal region. Here you can see through chalasa also. Here the entry is towards the through the chalasal end. From here it is entering. From the It is reaching towards, or maybe it should reach towards the egg cell. But entry, only entry, the entering part I'm saying, here it's directly entering through the micropylar region and moving towards egg cell. But entering through the chalaza land and again it's growing and reaching towards egg cell. So that type of entry through chalaza land, it is called as chalasogamy. Through chalaza land, chalasogamy. It's easy to remember. Through chalaza, the pollen tube is entering means it is chalasogamy. But it is not directly entering towards opposite cells. It is entering through chalasal end. That's all. Again, it's growing and entering towards the egg cell only. Okay. Here you can see the red colored marked one. Again, it's moving towards the synagogues and egg cell. And next type is mesogamy. What is mesogamy? The pollen tube. If the pollen tube enters through integuments, here you can see these are integuments, and through integuments the pollen tube is entering means it is called as mesogamy. In cucurbita and all you can see mesogamy. Through integuments the pollen tube is entering. Again the pollen tube, anyway it's wherever may be the entry part, if it is micropylar region or either it is in the integuments or chalasal region, whatever may be, again it will move towards the egg cell and synagogues. Here only the release, the content of that pollen tube is getting released near the egg cell only. Okay. So the pollen grains, if they are entering through integuments, they are called as it is called as mesogamy. Example is cucurbita. In cucurbita, you can see this kind of entry. And for chalasogamy, casuarina. Casuarina is the example for chalasogamy. So there are three types of pollen tube entry: porogamy through micropylar region, mesogamy means through integuments, and chalasogamy means through chalasal end. Clear? Next is so now we have already covered all parts of fertilization events. Formation of pollen grains we learned. Then egg cell, how egg cell is getting formed, all these things we learned. Then fusion of these two. Then formation of zygote. Up to that we have learned. So after zygote formation, whatever is there, whatever events are there, that is post-fertilization, because fertilization got over. After that, everything will come under post-fertilization events. Post-fertilization means after fertilization, the events after fertilization. So that's the end of fertilization events, right? Next, we are moving to post-fertilization events. First, let's see what are all the events that are in the post-fertilization. Here you can see endosperm development, then embryo development, then maturation of ovules into seeds, and maturation of ovary into fruit. So all these following events, all these are collectively called as post-fertilization events. So under post-fertilization events, what are all coming? Endosperm development, then embryo development, then maturation of ovules into seeds, then maturation of ovary into fruit. All these four events are coming under post-fertilization events. Okay. So in the post-fertilization events, the first thing or the first event is development of endosperm. Right. First one is development of endosperm. So endosperm, which is the product of triple fusion. We have already told that primary endosperm nucleus, that later it will give rise to endosperm. Right. So we have already told that. So let's see how endosperm is getting developed. So endosperm is a product of triple fusion. Note down, it's a product of triple fusion. It's not a product of fertilization. It's a product of triple fusion. So in NEET question, you may get confused. options will be there. So it is a product of triple fusion. So endosperm develops from the mitotic division of primary endosperm nucleus. So after the formation of primary endosperm nucleus, this will undergo mitotic division, not meiosis, mitosis. Okay, it will undergo mitosis, and as a result of that, that primary endosperm nucleus will form endosperm. So endosperm is a product of triple fusion, and primary endosperm nucleus will undergo continuous mitotic division and it will form the endosperm. And endosperm that develops from the central cell of embryo sac. In embryo sac, you can see it is 8 nucleated and 7 cell stage, right? We have already discussed this. Embryo sac means there are 7 cells. Here, thola cell and 3 cells, and the opposite also 3 cells. So on both ends, it will be having 3 cells, right? Here 3 cells you can see, here 3 cells, and the middle or the central cell is the large cell, right? So the endosperm develops from the central cell. Here the central cell, the nucleus of the central cell, that is the primary endosperm nucleus, which is in triploid condition, right? So that develops into, that endosperm develops from the central cell of the embryo sac. So from this large central cell only, that endosperm is developing. So it is a triploid tissue. We have already told it's a product of triple fusion. So it's a triploid tissue, and it is undergoing mitotic division. There is no meiotic division. Meiotic division means there won't be any equal sets of chromosomes. If it is getting divided meiotically, what will happen? See, there is 3 sets of chromosomes. 3 sets of chromosomes are there in primary endosperm nucleus, right? So that will never undergo meiotic division. Why? Meiotic division means reduction division. Exact half number of chromosomes will form after reduction division or meiosis, right? So this 3 sets of chromosomes, if it is getting divided, that will be some kind of abnormal cell. There won't be any equal division, right? There will be one and a half and 1.5 set of chromosomes. So that cell won't be a proper cell. Genetically, it will be having some kind of defects. That is not a proper division, right? So only this primary endosperm nucleus will undergo mitotic division. Only it will undergo mitotic division, and the cells formed a triploid tissues. Okay, so the endosperm development proceeds to embryo development. Means what? Endosperm, after the formation of the endosperm, or when it is after this endosperm development will start, only after that zygotic development. The development of embryo will start only after the development of endosperm, because this endosperm serves as a nutrient source, or that is the reserve food material for the embryo. If endosperm is formed or it is in halfway, then also embryo development will start, because only after the development of endosperm, this embryo can make sure that will get nutrients for the development, right? So the endospermic cells that will provide nutrients for the developing embryo. Got it? So the endospermic cells shows polyploidy. Polyploidy, because I said it's a product of triple fusion. So that will show polyploidy. So in cereals, you can see starchy endosperm. Note down, important MCQ for NEET. Starchy endosperm you can see in cereals, and in coconut, castor and all you can see oily endosperm. Oily endosperm you can see in coconut, castor and all, you can see the oily endosperm. That white part which is having the oil content. And in date palm, endosperm is made up of hemicellulose. Hemicellulose. Endosperm is made up of hemicellulose in date palm. Okay. So the endosperm development that leads to the development of embryo. Primary endosperm cell. Repeatedly, that primary endosperm cell will undergo division and it will form a triploid endosperm tissue. Which division it will undergo? It will undergo mitotic division and it will form a triploid endosperm tissue. So the cells of this tissue, they are filled with reserve food material. Which cell? The primary endosperm cell will undergo division. So the form, after that cells will be formed or tissues will be formed and these tissues are filled with reserve food material. This reserve food material is used by the developing embryo. So that provides nutrition for the developing embryo. And the primary endosperm nucleus undergo continuous nuclear division and it will form free nuclei. It will form free nuclei. Continuous nuclear division will take place and it forms free nuclei. That is called as free nuclear endosperm. Okay, the tender coconut water, that is a free nuclear endosperm, and the surrounding white kernel, it is the cellular endosperm. The tender coconut water, that is free nuclear endosperm because their nuclear division, continuous nuclear division is taking place. And when it is becoming mature, later the cellular formation will start. So that white kernel which is formed, that is cellular endosperm. So slowly when it is maturing, the cellular endosperm is getting formed, right? The white kernel, the cellular endosperm is getting formed later. So when it is maturing, the cell wall formation will start. So up to that, it is in a pre-nuclear endosperm stage. Then slowly the white kernel will get formed and that is the cellular endosperm. Okay, the embryo, forming embryo will be using the nutrients from this. In the starting stage, it will take the nutrients. The embryo will take nutrients from the tender coconut water, that is pre-nuclear endosperm. That pre-nuclear endosperm in the starting stage of embryo, that embryo will be using that one. And when it is developing, it will use the endosperm and that time the cellular formation will start. Got it? And next, endosperm. Here you can see tender coconut water. Here that coconut milk or the liquid endosperm, you can see that is the tender coconut water, that is the liquid endosperm. Okay, and the solid endosperm, the white kernel which is formed, that is the solid endosperm. And here this part, this is the coat of the fruit and seed coat, you can see the hard part, that is the seed coat. Okay, so the liquid endosperm which is present in the coconut water, that is in pre-nuclear stage. Okay, don't forget it is in pre-nuclear stage. MCQ, important MCQ. So commonly that primary endosperm nucleus undergoes continuous nuclear division and it is forming pre-nuclear form, and that endosperm development is called as pre-nuclear endosperm. And slowly that cell wall formation occurs and it will become later, it will become cellular endosperm. Okay, the tender coconut water is a type of pre-nuclear endosperm, important MCQ, and the white kernel present in the surroundings, that is the cellular endosperm. So don't get confused when they are asking about pre-nuclear, that is the tender coconut water, and the white kernel is the cellular endosperm. Okay, and next there are different types of endosperm: nuclear type, cellular type, and halobial type. Okay, three different types of endosperm. Nuclear type means in this the primary endosperm nucleus, Here the primary endosperm nucleus of the central cell. We know that central cell that divides without formation of cell wall. Their primary endosperm nucleus that will undergo division and there is no cell wall formation. This is the case where I told the free nuclear division. This is a free nuclear division. There is only nuclear division is taking place. There is no cell wall formation. So that is called as nuclear type. Okay. So the nuclei produced, they are free from the cell wall formation and they are freely floating in the cytoplasm of the embryo sac. So in the embryo sac cytoplasm, these nucleus will be freely floating. Okay. These nucleus will be produced after nuclear division and there is no cell wall formation and these nuclei, they are free in the cytoplasm of the embryo sac. And the cell wall formation, it may happen or may not be. Okay. In the case of cotton, maize, in all these nuclear endosperm. Okay, free nuclear endosperm. And second type is cellular endosperm. Here you can see the cellular endosperm. In cellular endosperm, primary endosperm nucleus. Okay, endosperm, the starting stage of endosperm is primary endosperm nucleus. So in all case, first the nuclear type means there only nuclear division is taking place. There is no cell wall formation. But in the cellular endosperm, that primary endosperm nucleus undergoes several cell division and each cell division, it is followed by cell wall formation. So cellular type means primary endosperm nucleus. Here you can see the primary endosperm nucleus. This will undergo division. But each division is with cell wall formation. You can see it is divided into different cells. There is cell wall formation in cellular type of endosperm. Next, so the endosperm, they have a cellular appearance in the cellular type. And examples, coconut. Coconut, first it will be in free nuclear form, then it will be in cellular form, right? You can see cellular type. Okay. And the third type is halobial endosperm. Intermediate between nuclear and cellular type. Okay, nuclear and cellular type. This is the intermediate type between nuclear type and cellular type. So first division, it is with cytokinesis. First, the primary endosperm nucleus will undergo division, and first division, first nuclear division, then cytoplasmic division. Both are there. Nucleus will get divided and it is followed by the cytoplasmic division. And after that, the remaining divisions. So again, after the first division, again it will undergo continuous division. But all those divisions are free nuclear. After that, only the first division is with cell wall formation. Remaining divisions and all, there is no cell wall formation. So in the first cell, you can see many nuclei here, and the second cell also you can see many nuclei here. So they, after the first division, it is free nuclear. So towards the chalazal end, here you can see the chalazal end, free nuclear division will be more. Towards chalasa, the free nuclear end. Here, this is the big cell. Here you can see more number of nuclei. So towards the chalazal end, the free nuclear division, it will be more. More number of nuclei will be seen towards the chalazal end. And in monocots, order halobials. Only in this you can see the halobial type of endosperm. Halobial type in the order, in one order. Which order? Halobial. Order halobials. Only in that particular order you can see this halobial type of endosperm. They are monocots. Under monocots, order halobials, you can see halobial you can see halobial type of endosperm. So there are three types: nuclear endosperm, example is cotton, maize; then cellular endosperm, coconut and petunia; then halobial endosperm, that members of order halobial, that is coming under monocots. They are not dicots, they are monocots. Okay. Next is embryo. So first endosperm formation got over. So I said after endosperm formation only embryo formation will start, because endosperm is providing nutrients to the developing embryo. So to make sure that nutrient availability only after the formation of endosperm or the development of endosperm, that embryo development starts, because it is assuring the nutrient availability. So the embryo develops at the micropylar end, right? At the micropylar end, that embryo develops, and only in that place the zygote is situated, right? The zygote, it is where the fusion of this egg and one male gamete is taking place, that is towards the micropylar region. So there only the formation of zygote is taking place. The zygote is getting divided and it is forming the embryo. So definitely the embryo development is also towards the micropylar end. So after the completion or after the complete development of the endosperm, or half, at least 50% of the endosperm should be formed, then only the division of zygote will start, because zygote need nutrients for the development of embryo, and this is a mechanism to provide nutrients to the developing embryo. Okay, if the endosperm is there, then the developing embryo can make sure that it will get the nutrients, right? And in monocots and dicots, the starting stage of embryo development, you can see it's almost similar. In monocots and dicots, the starting stage of embryo development is almost similar. Okay. Zygote, first it will form, here you can see. First the zygote will form the pre-embryo. Pre-embryo means just before the embryo stage. Pre-embryo. Then it will form the globular shape. Here you can see. Globular shape. Here it is almost round or globular shape. Then after some division, it will form heart shape. Here you can see a heart-shaped embryo. Got it? So embryo develops at the micropylar region, and after the formation of endosperm, only after the formation of endosperm, this embryo development will start. This is the mechanism to provide nutrients to the developing embryo. Okay. And endosperm or the embryo development in monocots and dicots, all the early stages in both cases are almost similar. And first, that zygote will give rise to pro-embryo. First, it will give rise to pro-embryo, then globular shape, then heart shape. Got it? Then next is next you can see here a typical dicot. We are going to explain about the dicotyledonous embryo. In dicots, the embryo consists of two parts. First is embryonal axis. Here you can see from this portion to down. This is called as embryonal axis. Then two cotyledons. This is two cotyledons. These are the two cotyledons, and this part is the embryonal axis. So typical dicotyledonous embryo consists of embryonal axis and two cotyledons. Got it? And next, in a typical dicotyledonous embryo, Just now I said we have embryonal axis, this part, and two cotyledons. We can see here two cotyledons. And the portion of embryonal axis. Here, this is the embryonal axis, and the portion of embryonal axis above the level of cotyledons. Above the level of cotyledon, it is called as epicotyle. What is epicotyle? Above the level of cotyledons. Portion of embryonal axis above the level of cotyledon, it is called as epicotyle, and epicotyle terminates with the plumule. Here you can see the plumule. Okay, above this part, that is called as epicotyle, and epicotyle terminates with the plumule or stem tip. This is the tip of the stem tip. Okay. Then the cylindrical portion below the level of cotyledon, it is called as hypocotyle. This is cylindrical portion below the level of cotyledon. These are two cotyledons, and below the level of cotyledon, the cylindrical portion below the level of cotyledon is called as hypocotyle. And this hypocotyle terminates its lower end. This hypocotyle terminates the lower end in radicle or root tip. Here, this is the radicle or root tip. Hypocotyle terminates the end at root tip or radicle. Okay, this root tip is covered with root cap. This is radicle means root tip. That root tip is always covered with root cap. So in diploid embryo, portion above the level of embryonal axis. Embryonal axis above the level of embryonal axis is called as, this is the embryonal axis. Above the level is called as epicotyle, and epicotyle terminates with the plumule or stem tip. Here, epicotyle is terminating. This is the epicotyle portion, and epicotyle terminates with the stem tip or plumule. And cylindrical portion below the level of cotyledon. This cylindrical portion below the level of cotyledon, it is called as hypocotyle, and hypocotyle terminates the lower end in radicle or root tip. This is radicle or root tip, the end portion. And this root tip is covered with root cap. There is a cap kind of thing here, which is covering the root tip. That is called as root cap. Okay. Then in a monocot, in a typical monocotyledon or monocotyledonous embryo, we can see that is having monocotyledon means that is having only one cotyledon. Mono means one, only one cotyledon. Dicot means that is having two cotyledons. And in grass family, they are belonging to monocotyledons, and grass family cotyledon is called as scutellum. Note down, important MCQ. All these terms are very important. Note down. You may get so many questions from this. Okay. So in monocotyledon, there is only one cotyledon, and in grass family, the cotyledon is called as scutellum. Here, the cotyledon, this is a cotyledon, a single cotyledon, that is called as scutellum. And the scutellum is situated towards the lateral side of embryonal axis. Here we said the embryonal axis, right? This is towards the lateral side, this towards one side of the embryonal axis, lateral side. So, the scutellum is towards the lateral side of the embryonal axis. And at the lower end of the embryonal axis, you can see at the lower end of the embryonal axis, radicle and root cap. Embryonal axis here also at the lower end, radicle and root cap is present. Same as in this case also, in monocots also, radicle and root cap at the lower end of the embryonal axis. And the root cap, it is, here the root cap is there in dicots. Here in monocots, the root cap, it is enclosed with undifferentiated shape called coleorhiza. Here at the end, radicle is covered by root cap in dicots. But in monocots, radicle is covered with root cap and the root cap is enclosed. Root cap is enclosed in a undifferentiated sheet. A sheet which is undifferentiated from the root cap. That is along with the root cap, that is called as coleorhiza. This is called as coleorhiza. And the portion of the embryonal axis above the level of attachment of scutellum. Above the level of attachment of scutellum. It is called as epicotyle. Okay. Here, almost it is the same. Here below radicle, root cap. And here also radicle and root cap. But one extra thing is coleorhiza, which is an undifferentiated sheet. That is called as coleorhiza. Then epicotyle has shoot apex. Here the epicotyle has shoot apex and leaf primordia. Shoot apex and leaf primordia. The epicotyle is having shoot apex and leaf primordia. And these, they are enclosed in a foliar structure. You can see here the foliar structure. It is enclosed in a foliar structure. That foliar structure is called as coleoptile. What it is called? It is called as coleoptile. So here lateral side scutellum is present and a foliar structure. Inside that shoot apex and leaf primordia is present. That is called as coleoptile. Then radicle and root cap at the downside. Radicle and root cap is present. And that root cap is with an undifferentiated sheet that is called as coleorhiza. Got it? And next, we can see seeds. Here different types of seeds we can see. And seeds, they are the fertilized ovule. After fertilization, it will form seeds. So seeds are the fertilized ovule. The ovule after fertilization, it will form seeds. So in angiosperms, seeds, they are the final product of sexual reproduction. Angiosperms, the final product of sexual reproduction is seeds. For the seed formation, all these fertilization procedures are taking place. So seed is a fertilized ovule. Got it? And seeds are found inside, where it is found? Inside fruits. And seeds are having seed coat and cotyledons. Then embryo axis, seed coat, cotyledons. First, seeds are having seed coat, then cotyledon and embryo axis. And the cotyledons, they are very thick and swollen, right? And the cotyledons, they are the simple structures and they are very thick and swollen. Why they are thick? Due to the storage of food. Got it? In, for example, in legumes and the cotyledons, they are thick and swollen because of the storage of food. And mature seeds, they may be non-albuminous. There may be non-albuminous or ex-albuminous seeds and albuminous seeds. Seeds are of two types, non-albuminous or ex-albuminous and albuminous seeds. Non-albuminous seeds are having, or non-albuminous seeds, that means there is no endosperm. There is no remaining endosperm. And during the development, the embryo will utilize this complete endosperm. They are called as non-albuminous endosperm. The endosperm is completely consumed by the developing embryo. That kind of seeds are called as non-albuminous seeds or ex-albuminous seeds. Example, pea, groundnut and all, they are the example for this. And albuminous seeds means that still some amount of endosperm it is remaining. There will be remnants of this endosperm as the reserve material, still it will be present. In maize, barley, wheat and all, you can see they are called as albuminous seeds. Albuminous seeds means residual of the endosperms. There it will be, or remnants or remaining part of the endosperm will be there as a reserve food material. In the case of barley, wheat and all, you can see that. And in some other type of seed, that nucellus, remnants of nucellus will be present. It is called as perisperm. Note down, perisperm. In black pepper, example is black pepper. In black pepper, you can see perisperm. Remnants of nucellus is also present in that. And next is integments of ovule. That integments of that ovule that will harden and it will form seed coat. Seed coat is formed from the integments. Hard integments will become hard and it will form the seed coat. The seed coat is having protective function. And micropyle of that will remain as a small pore. You can see here the micropyle will remain as a small pore. Integument will become the seed coat and the micropyle, that opening, it will remain as such, or it will remain as a small pore in the seed coat. Got it? And this helps for the entry of oxygen. It is a small pore, micropyle, it will remain as a small pore, and this helps in the entry of oxygen and water during seed germination. So during seed germination, through micropyle, the small pore which is existing, this micropyle, water and oxygen enters and the seed will germinate. So as the seed matures, the water content, when the seed is becoming mature, the water content of the seed reduces and the seed will become dry. Only the dry seed we can store, right? So the seeds will become dry and seed will become dry and the water content reduces. Only 10 to 15% of the water content will be remaining in the fruit. That seed will be in that stage. And moisture content will be around 10 to 15%. Okay. And metabolic activity of the seed, that also will reduce. And slowly that seed will enter into a dormant stage, or it enters into dormancy. And when it is getting water, or while sowing and while storing, it will be in a dormant stage. And when it is getting sufficient water and oxygen, or when it is getting a favorable condition, they will germinate. So the integments of the ovule that becomes hard and forms the seed coat. The seed coat is having protective function. Then the micropyle remains as a small pore on the seed, and this helps in the entry of oxygen and water during germination. And when the seed is becoming mature, it will become dry and metabolic activity also will reduce, and that enters into a dormancy. And when the favorable condition comes, it will absorb water, nutrients, and along with that oxygen through this micropylar region, and it will start germination. Got it? Next, fruit. Students, please wait another 5 to 10 minutes. Let me complete this chapter today. Next is fruit. That ovule. Here you can see ovule mature into seed. We have already learned. And that ovary is developing into fruit. Ovule is becoming the seed and the ovary develops into fruit. You can see. Here seed is there, seed coat is there, and the ovary, it becomes the fruit. And the wall of the ovary that develops into pericarp. Ovary wall that is developing into pericarp. This part, whole thing, it is called as pericarp. The wall of the ovary that develops into pericarp. And in some fruits, that part may be fleshy. In mango, orange and all, you can see flesh. That is a fleshy fruit. And in some fruits, we can see dry fruits, in the sense it's not like nuts and all. Not only that one, dry fruit like groundnut, mustard and all. There is no fleshy part. These seeds, they are the fruits, but they don't have the fleshy part. Okay, they are also a product of fertilization, but they don't have fleshy part like orange, mango and all. So ovule matures and forms the seed, and the ovary will develop into a fruit. And the wall of the ovary that develops into pericarp. Wall of the ovary. Ovary wall is developing into pericarp, and that is the wall of the fruit. Okay. Then this fruit may be fleshy fruit. Example, mango, orange and all. And fruit sometimes it may be dry also, like mustard, groundnut. Examples, you should note down all these examples. And many fruits, they have different types of seed dispersal mechanism. Many fruits have seed dispersal mechanism. Then in most of the plants, the fruits develops from the ovary, right? Most of the plants, the fruits will develop from the ovary. And while the development of the fruit, the other floral parts that may degrade or degenerate, or it will fall off, right? Other floral parts will fall off and the fruit will develop from the ovary. And the fruits developing from the ovary, they are called as true fruits. We can see true fruits which are developing from the ovary. You can see here true fruits which are developing from the ovary. And in some plants, fruits develop from other parts than the ovary. Not from the ovary, they will develop from some other parts. They are called as false fruit. For example, apple and all. This apple and all forming from the thalamus. Okay, the apple and strawberry, then cashew apple and all, it's not the fruit. Here, this is the fruit, that nut part, that is the fruit actually. This is false fruit. This nut part is formed from the ovary, not this apple part, right? In the case of cashew. And in the apple, that inside the seeds will be present, black color seeds will be present. That is formed from the ovary, not this fleshy part. Okay, so these are the false fruits. Got it? And in some case, some fruits without the fertilization, some fruits will form. They are called as parthenocarpic fruits. Without fertilization, some fruits will form. That is called as parthenocarpic fruits. Okay. So true fruits directly from the ovary, false fruit from other than ovary. Some other part will develop into fruit. That is called as false fruit. And parthenocarpic fruits means without fertilization, formation of fruits without fertilization. That is called as parthenocarpy. Next is apomixis. These are all important terms in this chapter. Apomixis means this is developed. The term is derived from Greek word. Apo means away from that mixing condition or mingling condition. That means the formation of plant from a seed without fertilization or normal sexual reproduction is not taking place. In apomixis, there is no normal fertilization. So it is very common in higher plants. In Compositae family, you can see Asteraceae or all these things you can see. Apomixis, there is no normal sexual reproduction. That means normal, there is formation of seed formation is there, but without fertilization. That is called as apomixis. Got it? Then in this, the diploid egg cell, it is formed without reduction division and without fertilization, it is becoming an embryo. There is no fertilization. Without fertilization, it is becoming an embryo, and the egg cell is forming without a reduction division. There is no reduction division, and without any fertilization, it is forming an embryo. That is happening in apomixis. Then polyembryony. Polyembryony is another condition. It is in that two or more embryos. Poly means many embryos. Polyembryony means many embryos will be there in a single fertilized egg. In a single fertilized egg, many embryos will form. Okay. Due to the embryos It is from the same egg and it will be identical to one another. Polyembryony, there is a condition of many embryos. From a single fertilized egg, formation of many embryos. That is called as polyembryony. That condition is called as polyembryony. And that embryos will be from the same egg. And that embryos will be identical to one another. And they are genetically, but they are divorced from the parents. That embryos will be genetically divorced from the parents. Got it? And next, the last part, how we are utilizing this apomixis in hybrid seed industry. What is the use of apomixis? So hybrids, how we are producing these hybrids? We are choosing male and female parent, different male and female parent with good qualities, and we are for mixing up of these characters, we are doing this hybridization, right? So hybrids are used for increased productivity. To increase the productivity, we are choosing the best characters for the male and female parents, and we are producing the hybrids. So the problems of these hybrids, what are the problems which we are facing from these hybrids? We have every year we have to produce the hybrids, right? Because from one generation we can't produce the next one. Again, genetically it will form different forms. So for a particular character, every year we need to cross. We need to cross the parents. Then only we can produce the same hybrid seeds. And hybrid seed production, it is not a cheap work. It is too costly, so that for farmers it is not affordable. So the hybrids are made through apomixis, means there is no segregation. The characters won't segregate. There will be the same character. The farmers can keep using the same seeds. Farmers can keep through apomixis. We are producing, means it is easy for farmers also. Otherwise, every year production, and it is too costly. It is not affordable for farmers. So that is the main plus point of this apomixis. In the hybrid seed industry, this is helping so much. It is helpful. So apomixis, then polyembryony, these are the two new terms. So that's all about the second chapter. Should I need to repeat anything? Is it clear? Any doubts in this part? Please respond. I need to repeat anything. Do you have any doubt? Deepa, then Guna Shelby. Is it clear? Okay, hope it's clear. There is no doubt in this chapter. If you have any doubt, you can ask me in the next class. And I have already given the questions from this chapter. Please go through the questions, practice those questions. That's all about second chapter. Next class onwards, we will go to the third chapter. Thank you. Thank you so much. You can leave.